want some salts in there as well. Does anybody know what kind of salts might be important? We talked about a metal that was required at the enzyme active site. Yeah, magnesium chloride. So here's what we need. In addition to those things, we need the template itself. Okay, but we need the polymerase, the DNTPs, magnesium chloride, and the primers. And then you're ready to go, as long as you have your thermal site. The primers, okay, so you need to be a little bit careful about how you design these. You want to flank the region of interest, so if you're trying to amplify, let's say, a particular gene, you need one primer that's like upstream, one primer that's downstream of that gene of interest. So it's actually kind of important that you know something about the sequence around where you're trying to amplify. The size of the primer is important. You need to get a long enough piece that it's going to be specific for that region that you want to amplify. And you want to have it be large enough, but not too large, so that the melting temperature, the annealing temperature, excuse me, is reasonable for the typical PCR thermal cycling. And you don't want one primer to have a completely different melting temperature than the other. It's important that they both get annealed that are right about the same temperature. So there are programs that you can use to calculate melting temperatures for different primers. And it's kind of interesting that you don't need the primers to be perfectly complementary to the region of the DNA that you're amplifying. You can have mismatches, and actually the incorrect sequence that's included in the primers will define your product, not the original template. And this just shows how you can use that to your advantage, right? So here is our template. We're going to come in with primers, but it's not going to be perfectly complementary in this sense. We're going to add a little extra spinach off the end of those, and those actually get replicated over and over and over again. So the product that you end up with in this case is the region of interest, but it also includes these additional segments on either end. And if we've designed our primer correctly, we can actually embed into them a site that's specific for an enzyme called an endonuclease that can come in and cut and leave these overhangs, which are really nice because we can use that to insert our amplified DNA into a plasmid or manipulate it in other ways. So right away, we're seeing how polymerase chain reaction can be used to benefit us and to introduce restriction sets for cloning. Now again, I'm going to sort of emphasize how we arrived at this technology, how it's like PCR did not come from above. Somebody actually went through this amazing inventive step, and it was this guy, Kerry Mullis. He was driving, I think it was a VW Bug, from Berkeley, where he was working, up towards his vacation place in Mendocino County, California. And all of a sudden, this idea occurs to him, and he pulls over to the side of the road, and he sketches out something pretty similar to that image that I showed on the first slide. It's like, wow, I should be able to make as many copies of DNA as I want. And it was just this thing that just hit him out of the blue. He describes it in his book. This is one of the first results that he published back in 1985 in the journal Science. And this just shows a very, very small region. Actually, this is like 8 nucleotide region that's been amplified. They were interested in amplifying a region of DNA so that they could evaluate whether or not people were susceptible for sickle cell anemia. So they were amplifying a bit of the beta globin gene in which the sickle cell mutation occurs. You may remember when we were talking about hemoglobin, we were talking about that mutation. So here's the original PCR report that allowed them to get this huge amount of these small oligos. They added the template, the nucleotide triphosphates, the primers, the buffers, the salts. They heated to melt the template. They cooled to anneal the primers. They added E. coli polymerase. They synthesized double-stranded DNA. They got a couple of copies of the template. They went back to here, and they repeated these steps. Cooled to anneal the primers, added more polymerase, got their copies, heated, cooled, added more polymerase, etc., etc. And this was pretty revolutionary, but it wasn't really what made this so powerful. Something here was missing that we just take for granted now all the time. And I'm sure, how many of you guys have done PCR? Wow, that's phenomenal. Probably a bunch of you did it in high school, I bet. That's incredible. Anyway, so this was the original report, but something's missing. What's missing from this? Didn't they provide that makes this kind of a pain to do? Yeah. They have the template around, yeah. So that wasn't the issue. So I'll give you a hint. They're repeating this step over and over again. They're adding polymerase every time they cycle. What's going on?
Is it tag polymerase?
They left out Taq polymerase. What's so special about Taq polymerase? Is Taq like so wonderful that E. coli is not so good? E. coli does a great job. We talked about E. coli polymerases. Yeah. Exactly. E. coli polymerase craps out. So when you heat here, you kill your polymerase, and that's why you have to add it at each and every cycle. Why doesn't that happen for Taq polymerase? Yeah. Exactly, and I'll show you a picture of that in a bit. So here's the original paper, okay, published in '85, and there are people like, oh, that's kind of interesting. This is the number of citations for the paper, and this is kind of the way that you can judge the impact of a paper. Like, okay, so here's, after a couple of years, it's getting cited like over 100 times a year. 100 citations for a scientific report is phenomenal. I'm really happy when I get a paper that gets cited 100 times. After a few years, it's being cited 700 times or more. You can see people got used to it. Now people kind of take it for granted. So that was the original report, but when they published their update with the Taq polymerase, now you can see that it's getting cited almost 2,000 times a year. And look how quickly that this idea caught on. Like, they published in '98. Within like 2 or 3 years, everybody was like, okay, this is fantastic. This is exactly what we have to be doing. So that was a huge deal. And then you can see from the name of this paper, primer-directed enzymatic amplification of DNA with a thermostable DNA polymerase. That's what made it convenient. They didn't have to pry open the stupid machine every cycle and dribble in more polymerase. They just set everything up in the beginning, shut the door, and then it was programmed to go over and over and over again. I actually worked in a building where Kerry Mullis did his first experiments on this, and they showed me to some, I was some like schlumpy grad student. They showed me to some like room with a whole bunch of just dusty equipment. I said, okay, you can work in here. I was like, okay, well, I need to clear some space to do my experiments, and here's like these incubators. I'm going to get rid of them. They're like, no, the Smithsonian wants those. Like, why would the Smithsonian want them? This is the first incubators that were used to do Kerry Mullis's thermal cycling. All right, so it's a special polymerase that we ended up using, and it comes from this organism, Thermus aquaticus, and it's Taq polymerase. And this thing grows in hot springs. Its optimum temperature is 75 to 80 degrees. So yeah, you can heat this thing up 80, 90, 95 degrees. That polymerase will remain folded. It's got a half-life of minutes, even up at 97 degrees Celsius. And it's pretty speedy. It can replicate 1,000 base pairs in less than 10 seconds at 72 degrees. So we can get each of these cycles to happen really quite quickly within a period of a few minutes, and you can rip through 30 cycles within a period of a few hours. The original Taq polymerase has no exonuclease activity. It doesn't have proofreading, so the fidelity was not that great. An error rate of 1 in 9,000, but actually if you're only amplifying a region that's a couple of thousand, that's probably good enough. So this was all right. There's a lot of other polymerases that are now available. And this just shows this beautiful picture of some of these hot springs where these kinds of organisms grow. Excellent. All right, so now is my segment for Meet the Chemist. And I'm going to talk to you about this guy, Zach Charlotte Powers, who's one of my former students. He took my biochemistry class a number of years ago. And he now works for some biotech company here in New York, I think. But what I'm going to talk to you about today is the fact that he's not only a scientist, but he's also a rapper. And so this is so goofy, I have to show you this.
Let's do this. That's awesome. They actually have 2 songs. I didn't know you rap. I hear your music, bro.
Let me tell you a little story that you might like to know about a biologist. He made a lot of dough. He perfected a technique to copy DNA. He patents it and lives in a mansion today. His name is Carrie Moses. He's a PhD, a regular Joe, just like you and me, except he figured out how to go real far. He's the inventor of a technique called PCR, polymerase chain reaction. It's a fancy name for the way things happen when you take a double strand of DNA. Copy it exponentially. This method is simple, but you must pontificate on the mechanism by which DNA replicates, and therefore a review of its structure is necessary. The double helix won't be forced down Harry. You can isolate naked DNA in the tube. Anyone can do it. It's not hard to do. We take advantage of its physical properties. Hydrogen bonds between the strands in the backbone and covalently. Each strand is made of 3 smaller parts: a sugar, the backbone, a choice of nucleotides, and the sugar phosphate is the skeleton of the molecule. The nucleotides are the bearers of its goodwill, because it is the A, C, T, and G that will base pair in a manner complementary. The hydrogen bonds between A and T, C, and G is the basis of the helix in which is also an entry, because we will melt the molecules with heat. Counter-free nucleotides we include in the heat to line up correctly with the counterpart on the strand. This will form the basis of our copying plan. As we heat the DNA, hydrogen bonds separate, cool it down, and they reform piece of cake. But if we put a primer into our reaction mix, when the DNA cools, the primers will affix themselves to the DNA at their hydrogen bonded places, acting to start catalysis of polymerases, because the hydrogen bonding of nucleotides with each new copy templates the other side. So every heating cycle yields a copy of each strand, and double geometrically expand. 2, 4, 8, 16, 32, 64, 122, 56, 5, 1, 2. For 30 cycles we can make 1 billion copies. This is the reason why Carrie Moss is called pop. Oh baby, you double-stranded DNA. Your helix I will cleave if I give you enough heat. Oh baby, you single-stranded DNA with heat-activated enzyme catalyzed from 5 to 3 prime. This sort of probing can let you take the smallest of the smallest sample of DNA with PCR and a few cells. You can begin to look inside that human genome, that beautiful book of life it is living. And I would be fibbing to tell you to society it doesn't give back, it is giving. Every day PCR is used in math. Hospitals, researchers, it helps in their task. And every time they use it, because it is licensed to give a little money to our man, yet to him, a graduate student turned millionaire. Using his nuggets, he's got a plan in his Ferrari. Now you'd be sorry that you didn't study biology.
He's done a few of those, I think. Great spirit. Yeah, I don't think anybody's calling Carrie Mullis Poppy. I can't be sure, but I don't think he's driving a Ferrari. He did get the Nobel Prize, which is pretty cool. All right. Some of the applications that were alluded to in that video, it's pretty amazing. We can now test for genetic diseases. That's the very first thing that they were trying to do there. Amplify DNA for testing sickle cell anemia. Tissue typing, blood typing, personalized medicine, trying to find out what mutations might be driving cancer, disease diagnosis, forensics. Yeah, now they can obtain enough DNA from a fingerprint to figure out who somebody is. I think that's the way they caught that bomber who was recently sending bombs from Florida. Crazy guy. Parental analysis, DNA cloning, site-directed immunogenesis. Oh, some really great work done in anthropology, cryptozoology. There's this guy here, Todd Dissotel. Has anybody heard of this guy? He's over in anthropology. He's got this TV show. He goes around the Pacific Northwest and he collects samples that people think is from Yeti, and he tests to find out whether or not there's an actual Sasquatch. Really interesting, very amusing work. And analysis of gene expression. We now have reverse transcriptase PCR so that we can quantitate how much of any particular gene is being created. So many different applications. Zach himself ended up doing some really cool work with PCR. They went to different places around New York Harbor and just pulled water out of the harbor and amplified fish DNA, and then were able to do enough sequencing to figure out what type of fish were present in New York Harbor at any particular point in the year. And so they were able to figure out, okay, when are the bluefish showing up? When are the anchovies arriving? And they also found that there was a very small amount, about 2% of fish that were not endemic to New York. It's like, where is that coming from? People are going to the store and they're buying fish, and when they are cleaning it, they're putting it down the sink and it's ending up in New York Harbor. And this technique is so sensitive that they're able to amplify the DNA from New York Harbor and figure out what people are eating for dinner on the Upper West Side. Pretty incredible. And they can do that because the cost of sequencing any particular genome has dropped phenomenally. And a lot of you have heard of Moore's Law, which is about the cost of computing and how it relentlessly decreases, making our computational power that we can hold in our hands and afford increase from one year to the next. Here is sort of the extrapolation of Moore's Law. This has been going on for a couple of decades now. But look at the price to sequence a human genome. Right around the time when the Human Genome Project got started, we're talking tens of millions of dollars to do one human genome. And then this drastic drop in price, 100K, 10K, and as of 2016, it now only costs about $1,000 to sequence an entire human genome. It's amazing. It's a faster drop in price, faster increase in efficiency than even Moore's Law for computing. Fantastic. Last year I was back in the Bay Area. I look up in the sky and there's this blimp, 23andMe. It's like, oh my goodness, sequencing really has come a long way, but sequencing companies have their own blimps now. But yeah, you can get, obviously, probably a bunch of you have had. How many of you have had your sequences identified, your genealogy? So that you do it. You can also do parental analysis. Here's this woman. She claims that Keanu Reeves came to her at some point and she got pregnant from him. She claims that he disguised himself as her ex-husband and sued him for paternity and parental support. She was suing him for $3 million per month. And fortunately, we have PCR, so we can provide the DNA analysis, and Keanu won that case, and so I give him $3 million a month in spousal support. All right, but why should you guys care? Well, it's really important because actually this sort of technology is allowing the development of new pharmaceutical agents. We can use PCR and site-directed mutagenesis to come up with recombinant DNA techniques to create protein therapeutics, including insulin, growth factors, erythropoietin, all kinds of growth factors and vaccines as well. So really important. For me, this has become really crucial, the use of PCR in forensic analysis, because I've gotten to know a lot of these guys, well, a few of them anyway, who were exonerated for crimes they didn't commit based upon PCR amplification of DNA that was left at the crime scene. And that evidence was used to definitively prove that these people were incarcerated and found guilty for crimes they absolutely could not have committed. This guy, Alan Newton, served from 1985 to 2005. And his lawyers began asking New York City for DNA that was at the crime scene. For 10 years, New York City refused to provide it. Finally, somebody took the time and effort to dig this stuff up. Small amount of material, used PCR to amplify it, sequenced it, definitively showed that this guy couldn't have done the crime. He was exonerated after serving 20 years for a crime he didn't do. It's amazing. He got a job at Medgar Evers College. He's been an inspiration for a lot of other people, and this just keeps happening over and over and over again. There's an organization here in New York called the Innocence Project. That's what they do, and they've been able to go back to samples, material that was left at crime scenes, and just get hundreds of people now out of prison for crimes they didn't do. And this guy, George Allen, 30 years behind bars in St. Louis for a crime committed. I mean, when I first heard about this, I thought maybe a few dozen people, but it just keeps going. This organization keeps finding more and more people to exonerate. So, power of PCR-driven amplification. All right, any questions about PCR and its applications? Yeah.
Allow the primers to enable to the individual temperature.
How does the polymerase know when to stop when it's propagating from one primer in one direction? The answer is it actually doesn't. So it'll continue going, but at some point you change the temperature. So it's only got a couple of minutes. It's not an infinite length. And the issue about why does it only amplify that particular region is that it's only really going to be efficient at amplifying based upon where the primers are. So as you get going initially, yeah, you'll get some that have larger extensions, but what ends up as your product is going to be defined by the primers on each side. Okay. Yeah?
How do you actually get a primer that's appropriate for what you can do? They sell them?
Yeah, but you can easily get primers that are typical for common genes, like let's say mitochondrial DNA. And these days, if you have a sample, you can submit your sample for amplification and sequencing, and they will actually generate the primers for you. It's just super easy. You just have to give them the sequence. They generate the primers and include them in the amplification mixture. Yeah?
What happens if you only use one primer?
You would end up getting affected by this issue that you would keep going in one direction and you'd get a heterogeneous amount of material that would not be very well defined. You'd get a bunch of copies, but you'd only really be able to be amplifying one of the two strands, because one of the primers anneals to one strand, one of the primers anneals to the other strand. So you'd only get one strand amplified, and it would be poorly defined as far as where that would end.
What if like the sequence is close to the end of the DNA so that you can, it's like, if you have the correct extension time.
No problem if the sequence is primed towards the end of the DNA. I showed you one example there where they only amplified like 8 nucleotides. I mean, whatever's there is going to get amplified. And so, yeah, if you wanted to set up your primer close to the end and just amplify that bit, and you wanted single-stranded DNA for some reason, yeah, you could do that. That would work. All right. Anything else? Good. Let's now talk about this second step in the central dogma. Okay. So we've pretty much covered this issue, DNA templated DNA synthesis. Now we're going to move towards transcription, DNA templated RNA synthesis. And this is crucial because this is at the heart of gene expression, right? When we express a gene, it means that we're generating an mRNA version of the gene that's encoded in the DNA genetic material. So I'm going to ask this question: what is a gene? And I want you to really think about it. Professor Dow, do you have a good answer for what a gene is? Do you think that you have some kind of intuition for what that 4-letter word means, gene?
It's something that that is that carries out the information of a person.
Okay, great. So it's a unit of inheritance. It's a unit of what defines what a particular individual's phenotype is, right? And we must have many units because we have different aspects of our phenotype. We have what defines our hair color, our eye color, that sort of thing. So yeah, that's the very first definition that people began working with. It's a part of the chromosome that defines a phenotype. But that's not too satisfying on a molecular level, and so more precise definitions come about. That what creates a phenotype? Well, different enzymes very often are what really define the phenotype. So we had this notion that, well, there's one gene, and it's going to create instructions for generating one enzyme. And then we got a little bit more precise there. Okay, well, we need a message. So one gene creates a particular messenger RNA, which then defines one protein. And so, yeah, not all proteins are enzymes, so that's helpful. It's moving along. And then it's like, well, it's not always a full-length protein. Sometimes it's a polypeptide. So this was like kind of a subtle variation on that definition. One gene encodes one mRNA, which makes one polypeptide. And then there was an appreciation, especially within the past couple of decades, that, well, actually, we're making RNA molecules, and sometimes those carry out really important functions within the cell. So it's not just protein polypeptides that are being generated from genes. Sometimes it's particular RNA. So then it's like, okay, well, one gene makes one gene product. And now that definition doesn't sound too valuable. It sounds kind of like a tautology. It's like sort of just referencing itself. But that's where we are. And so we're still struggling with this definition of a gene. I just think it's incredible that we've been talking about genetics and the power of genetics in modern society, but we don't really have a good definition for what a gene is. Here's what your textbook says. Genes are functional units of DNA. So this is what a biochemist would say, biochemistry textbook. Functional units of DNA are defined by the RNA products they produce, which in most cases refers to mRNA products coding for proteins. I don't have any problem with that definition. That's probably the one that I work with a lot. Just operate intuitively. But it is kind of waffling here, in most cases. And I really enjoy following the debate in the scientific literature about whether or not we can actually provide a precise definition for what a gene is. This is one of the more recent papers that I took a look at, and it says right there, this is from the journal Genetics, the present period features complexity and confusion regarding the definition of this term. And they provide their own suggestion for what the definition should be. A gene is a DNA sequence whose component segments do not necessarily need to be physically contiguous. It specifies one or more sequence-related RNAs or proteins, okay, so that's good, that are both evoked by gene regulatory networks and participate as elements in those networks, sometimes with indirect effects or as outputs, yielding more direct phenotypic effects. So we're getting a little bit of the flavor of the fact that genes provide phenotypic effects, but it's filtered through a particular molecular definition, right? We're talking about DNA sequences here. So this is okay. It seems, I don't know, not intuitive, a little bit jumbled, but I just really enjoy following the debate. I think it's fascinating. This part right here, segments do not necessarily need to be physically contiguous. So that's kind of an important point here. Right? We used to think of very well-defined regions on our chromosome. Okay, this particular region defines a gene, and then this is pointing out that it doesn't need to be one isolated region. And I brought this up last time, that genes are in pieces, right? And this is like kind of best case scenario. In eukaryotic systems, typically when we have a gene, it doesn't just encode for the particular mature mRNA and the particular protein sequence. It's got these intervening bits in them, called introns, and those are non-coding portions of these genes. And they need to be removed. We'll talk about processing of immature mRNAs and how these introns are spliced out next week. But you can see sometimes these introns are huge, and sometimes the intervening regions, the non-coding regions, are actually longer than the coding regions, which is the case here in hemoglobin beta subunit. What is the function of these introns? Well, for sure that in many cases they play regulatory roles. I'd say a general description of what the functions are of all introns, it's not really apparent. But we do see huge amounts of human genome does not actually encode for any functional protein. And in fact, this quote, I think, describes the situation pretty well. The relative paucity of genes in the human genome leaves a lot of DNA unaccounted for. Yeah.
So genes can be discontinuous, but would you ever see a gene that spans multiple chromosomes, or just within one chromosome is broken up?
Lately, I've been reading about these huge discontinuous things. I even think I've seen that some genes are encoded partially in the nucleus and partially outside of the nucleus. Again, depending upon how you might define a gene. I don't know if anybody has seen any reports about that. But yeah, no, distances between different components of a gene can be substantial. All right, a lot of DNA unaccounted for. We talked about how big the human genome is, millions of base pairs, but really not that many more genes, only 20,000, than much simpler organisms. And this just shows how much region of the human genome is not transcribed at all. This green bit is protein coding. You see E. coli is very efficient, almost the entire genome codes for protein. Yeast is still pretty efficient, but wow, by the time you get to humans, it's just a tiny little sliver. And we've got these other non-coding RNAs that are verified. We know that an RNA product is generated In this case, we believe an RNA product is generated from the DNA template, but there's a whole, almost half of the human genome we believe is not transcribed at all. So the question of what that's doing there is, I think, also an interesting debate. All right, but let's talk about RNA. So what types of RNA are there, and what do they do? What kinds of RNA do you guys think about? Yeah. mRNA, tRNA, good. Those are two of the biggies. There's another biggie. Okay, ribosomal RNA, and we'll take a look at the RNA that's used to form part of the ribosome, the protein synthesizing machinery. Okay, so those are the three big ones. There are other RNA species. We'll talk a little bit about what those are. But RNA plays a lot of roles. It does form information storage, right? It carries the message from the DNA to the ribosome. It's a regulatory molecule. They're short interfering RNAs that regulate the expression of genes. We'll see that RNA plays a catalytic role in the cell, and it's also a kind of a structural polymer. It's used to build up the ribosome. So here's a table from your textbook describing the different types of RNA. These are the ones we typically think about. And then there are these non-coding RNAs that are segregated in what they call short, small, and long. Some of these I'm pretty familiar with, and I hope we get a chance to talk about them when we talk about gene regulation, microRNAs, short interfering RNAs. But I'm going to be totally honest with you guys. Like, this is a new textbook for me. I just started using it this year. I'd never heard of some of this stuff. Turk RNA? I think I've heard of Pee-wee interacting RNA, but I'm not quite sure. So all of this is pretty new, and at least in my mind is still somewhat obscure. So lots for me to learn about with regards to what RNA is doing in the cell. But as we contemplate the central dogma and figure out how the information passes from DNA to RNA to protein, we will be focusing on mRNA, tRNA, and ribosomal RNA. So these show different genes, and we oftentimes really focus on the protein coding genes, and those are the ones that crank out an mRNA message that's brought to the ribosome and is used to provide for the information about which amino acids should be added in which sequence. But in addition to that, there are genes that encode for the tRNAs. These are the tRNAs that bring the amino acids to the ribosome. And then, as I mentioned, the ribosome itself is composed of different RNA molecules. So we have these different types of genes and different polymerases that are actually used to make mRNAs versus tRNAs, for example. This is another kind of representation of the central dogma. And today we're going to really be focusing on RNA polymerase. But the point that I'm making here is that we think a lot about template-directed linear polymer syntheses as we go through the central dogma. And the main ones are these ones here in yellow: DNA-encoded DNA synthesis, DNA-encoded RNA synthesis, and RNA-encoded protein synthesis. And we will talk briefly about different ways to get information to flow from RNA back to DNA using an enzyme called reverse transcriptase, and also that in some cases, RNA serves as the repository of genetic information for some virus, some viral entities, and in those cases, there is such a thing as RNA-dependent RNA synthesis, and those polymerases exist too. These green ones are the oddballs. We'll just be mentioning them quickly, and we're really going to focus in on these three types of polymerases. I'm emphasizing this aspect again, the fact that you should know for all these template-directed syntheses, what's involved in initiation, what's involved in elongation, have an understanding of how those elongation events are processive, that the polymerases sit down on the message and they just continue to work on it. They don't diffuse on and off at each step. And then also that each of these polymerizations needs to be terminated at a particular position. I'll also just introduce this as kind of parenthetically that reinitiation events can occur. We can actually be using a particular template more than once so that we can accelerate production of the product if we need to do that. All right, here's the protein machinery that we're going to be focusing on today. This is the RNA polymerase, and here are two different flavors of that. One is the polymerase that's coming from a prokaryotic system, bacterial, and this is the yeast RNA polymerase, so from a eukaryotic system. This is a multi-protein piece of machinery, and we'll be talking a bit about the different roles of these different proteins, in particular, role of the sigma factor. When we bring all these different subunits together, we call that the intact holo enzyme. And the eukaryotic version, sure, it's a little bit more complicated. It's got a few more protein subunits, but you can probably already see that it's got some of the basic architecture left intact, and there are a lot of similarities. Just as in the case of DNA polymerization, though, we'll be focusing in on the prokaryotic system. It's easier to understand, and the basic elements are what's really crucial and can be appreciated most readily by looking at the bacterial system. So let's take a look at the RNA polymerase from E. coli. And just as we were talking about for polymerase chain reaction, I want you to understand what components are required to make this polymerization happen. So we need our holoenzyme, this guy. We need our template, just as in the case of PCR. We need our substrates, the 4 ribonucleoside triphosphates, right? And so we'll be using ATP, GTP, CTP, and because we're making RNA, UTP. And then we also need magnesium, just as in the case of PCR, because this polymerase also has magnesium that's present at its active site. Yes?
Can you explain the need for magnesium?
I will in a few slides. Okay. Here's my question for you guys. What reaction is being catalyzed? I'm going to make this really easy. It's essentially the same exact reaction that I asked you to know when it came to DNA polymerase. Yes. Which hydroxyl? The 3rd hydroxyl. The 3' hydroxyl? Great. So the 3' hydroxyl on the growing RNA strand is going to be a nucleophile. What is that nucleophile going to attack? The 5' phosphate. These are triphosphates. Which phosphate? First one? Okay. The alpha phosphate. From the 3' end of a growing RNA strand, nucleophilic attack of the 3' hydroxyl at the 5' alpha phosphate of a ribonucleoside triphosphate. Fantastic. You should be able to draw that arrow. And as you're drawing out that reaction, you should also represent the other product, which is going to be what? After the nucleophilic attack, We're going to extend our polymer chain. We are going to release pyrophosphate. Excellent. Beautiful. So we've got the chemistry down, and I'll show you what the active site looks like in a bit. Prokaryotic RNA polymerases, they've got their core enzyme, and they've also got an initiating sigma factor that floats on and off to the machinery. We'll talk about the importance of this. The core has got 4 protein units in it, 2 copies of the alpha subunit, a beta and a beta prime. That's a tetramer. That's got the catalytic subunit in it, and its overall molecular weight is about 500,000 Daltons. Pretty hefty. Its mechanism, as I've been emphasizing, is similar to DNA polymerase, but there's no proofreading, and we went into what goes on as far as proofreading with DNA polymerase. RNAs generally are not corrected. The error rate ends up being about 1 in 10 to the 5, which is more or less what you would get from DNA polymerase if it didn't have the proofreading and exonuclease activity. So, I'm going to ask this again. I asked it before. Why don't we care about RNA being having that same type of fidelity as the DNA polymerase? Right. Exactly. And oftentimes we can readily tolerate a particular error in an mRNA, and it will actually generate a functional protein. So what's the sigma factor? It's not part of the core enzyme. The sigma factor finds the promoter sites where to begin generating the RNA, and it kind of scans through the DNA, the genetic material, looking for places to initiate RNA synthesis. And so it actually is a 1D diffusion. The sigma subunit is like sitting on the DNA, and it's searching for the start site. This shows the holoenzyme, right? This is the core, and here's our sigma factor right here, is this pink blob. And you can see that it's sitting down on the promoter region. This promoter is a sequence that defines where we want to begin our RNA synthesis. And there's 2 particular regions that are about 10 base pairs and about 35 base pairs upstream of where we want to start the mRNA synthesis. And that's what that sigma factor has an affinity for. It likes these rich regions. And so it'll just scan through until it hits it, and then it will associate with that, and that directs the holoenzyme to begin its process at that position. So transcription starts with binding and initiation. Okay, so that is something that we need to keep in on, right? So the sigma is really defining where this is going to associate on these upstream regions. Once the holoenzyme complexes with the duplex DNA, it generates an open complex. What do we mean by open complex? That means we can't do a good job of creating an RNA copy if the DNA is present in its duplex form, right? We need to pop it open, get access to the bases so we can line them up and make the proper hydrogen bonded complement for the incoming RNA trinucleotides, nucleotide triphosphates. So a conformational change happens here, and it generates this opening in the duplex DNA. And once that open complex is created, it can initiate transcription, begin its polymerization. It doesn't use a primer in this case. And that sigma factor then can be released. It's done its job. The sigma diffuses away, and the core units, those catalytic units, begin their process of cranking out the RNA product. Questions about that? Yes?
Why do we need to generate 8 or 9 bases before the sigma unit?
I don't know that you would need to in a theoretical sense. I mean, I guess once you initiated the open complex, presumably the sigma complex could lead right then and there. That's just what's been observed. That's what happens, is that a few bases are polymerized before it does diffuse off.
It just takes time, or like there's no known purpose if you take temperature?
Kinetics, the timing of these events does turn out to be important, both with regard to initiation and with regard to termination as well, as we'll see in a bit. Yeah. How do we study these sorts of interactions? How do we know where a sigma factor will bind a promoter region? How do we identify promoter regions? How do we identify in a general sense where a protein might engage a DNA as a binding partner? We have three main techniques that we can use. We can allow a protein to associate with DNA and then just subject it to a digestion process, chemical digestion, cleaving off the DNA. And generally what happens is that you really do a great job of digesting the exposed naked DNA, but where it's complex with the protein, it's protected, and then you can try to do a decent job of sequencing that bit that's been left behind. Second technique that your textbook describes quite nicely is a footprinting experiment, where you compare digestion using a nuclease that clips, this is an enzyme that clips the double-stranded DNA, and you compare the digestion pattern in the absence and in the presence of particular proteins that you suspect are binding at very particular sequences in the DNA. What happens? That enzyme, the DNase, nuclease, it will cut throughout the naked DNA, kind of randomly. We're looking for a situation. It makes, on average, one cut, and that generates a ladder of all the different sizes of DNA that could be made for cleavage all the way along that substrate. We repeat that experiment in the presence of the binding protein, and now at those sites where the nuclease was able to cut before, that enzyme can't get access to the DNA because that's where the protein is sitting. And so when we run a gel electrophoresis to visualize the bands of different molecular weight products that are generated from this process, we see that a couple of them are missing. Those are the regions that have been protected from digestion by the presence of the protein. And that helps us define the sequence, right? So this is allows us to identify promoter sites, and this is what a typical experiment might look like. That if you digest it in the presence, in the absence of a protein, you get all these different bands, and then if you do repeat the experiment in the presence of a protein, in this case, it's binding to a lac promoter. We'll talk about that a little bit later. You see that the cleavage is protected where the protein is bound, and have these missing bits in this ladder that shows us where the protein is binding. And when we look at the different sequences that are bound by different, that define different promoters upstream of different genes, we see that there are these repeating patterns, these consensus sequences right upstream of the initiation site, these so-called Pribnell boxes, and these correspond to conserved regions that are about 10 to 35 base pairs upstream of where we want to start making our mRNA. And finally, the third experiment that's often used is called the gel shift, and that just looks for a complex that's formed between a protein and DNA, altering the molecular weight of that complex and shifting its position in a protein electrophoretic gel. All right, so let's get into the nitty-gritty of what the polymerases are doing. This shows the RNA polymerase, the holoenzyme, bound to the DNA. We see the sigma factor is down here. Here's the DNA that's being replicated, and you can see that there's partial separation of that DNA into two different strands. So the sigma factor really is a regulatory element in bacteria. It will help define what genes are being expressed at any point in time, depending upon which sigma factor is being associated with the holoenzyme. And different sigma factors will bind different promoters. So here's a standard one. And then there's different upstream promoter sequences that help define genes that need to be expressed under particular circumstances. So here's a sequence that's upstream of, let's say, heat shock genes. These are genes that the organism needs to express in the presence of elevated temperatures or in the deprivation of nitrogen. So this is the way that whole sets of genes can be turned on and off very quickly just by switching out which sigma factor is being used by the cell at any point in time. This is a modular unit. This is the sigma factor right here. I'm sorry, this is alpha factor? It should be the sigma factor. Anyway, this shows the RNA core polymerase, and this shows that we're opening up the duplex DNA, and we're creating like a bubble, and it's that single-stranded DNA that's being fed into the enzyme active site, and there's our site of catalysis. So here's a schematic of what that looks like. We've opened up the double-stranded DNA, two single-stranded regions, and then one of them is described as a coding strand. It's actually the other strand that's the template strand that's being used to line up the incoming nucleotide triphosphates. There's a channel that those nucleotide triphosphates come into, and then they hit the active site and they're added to the growing 3' end. The RNA is fed out of a separate exit channel. Here's our product coming out, and then the DNA duplex is reformed, and the double-stranded DNA also has its own exit channel. And the polymerase just continues to crank along, opening up the double-stranded DNA and forming the product. All right, so what's the magnesium doing? It's playing a very similar role here as it was in the DNA polymerase. Two divalent cations, generally magnesium, complex with 3 aspartate residues. This magnesium is sitting right near the 3' hydroxyl, and it's activating that oxygen for nucleophilic attack. That nucleophilic attack is being conducted on the alpha phosphate of the incoming ribonucleoside triphosphate. All right. This shows the sequential steps. I talked to you before about the generation of the open complex at the gene promoter, the fact that the sigma factor is released, the polymerase carries out its synthesis, moves down the DNA template until it's ready to terminate. So let's conclude this with an understanding of what drives the termination of this polymerization process. Here's our transcription bubble. We're unwinding the DNA ahead of the synthesis. We're rewinding it behind. And there's 2 types of termination signals that are typically used. One is when the polymerase finds an unusual structure that's been generated within the RNA. If it generates this kind of product that is got 4 U's, 4 uracils in a row, in proximity to this hairpin structure that the RNA product can use, this serves as a signal for the polymerase to stop and disassociate. There's also a protein factor that can be used to tell the polymerase that it's done. So here's our hairpin, and then the polymerase undergoes an isomerization event, it stops, and then can disassociate from the template. And then there is the use of this Rho factor. This is the other mechanism for termination. And the Rho factor is basically chasing along behind the polymerase, and there are a bunch of different possible sites that it can signal to the polymerase, okay, you're done, stop doing what you're doing. And in this case, the polymerase may stall at different points as it undergoes a variable rate of movement down the DNA template. And when it stalls, the rho factor catches up to the polymerase and basically pops it off the template. And it's only because the polymerase varies in how fast it moves down the template that there are particular sites where the rho factor is typically going to terminate the RNA synthesis at particular positions. Here's our rho protein. It's actually ATP driven. Chases after the polymerase. It's hanging on to the RNA product. And when it hits the polymerase, when it slows down, it tells the polymerase to terminate. So the rate of elongation, the rate of RNA polymerization is variable. Certain sequences in the RNA and in the DNA template slow down the polymerase and allow the rho to catch up and terminate. We'll talk next time about additional protein factors that help regulate transcription.